In 1976, a virus appeared in a small village in Central Africa. It killed most of the people it infected, and then it vanished. Not slowed down, not contained. Vanished, with no cases anywhere for years. Then it came back. Then it disappeared again, and it has done that for 50 years. In all that time, with everything we have built, scientists have never found where it goes. And that is not a figure of speech. Nobody has ever pulled live Ebola virus out of a wild animal, not once. We have vaccines for this thing, and we still do not have its address. Right now, it is spreading again, in a form our vaccines were never built for. But before we get there, you need the 50-year manhunt. Because when scientists started finding where it hides, the answer was somewhere nobody had looked. Hit subscribe and ring the bell, because this one gets strange. First, forget the movie version of this disease. You are picturing someone bleeding from the eyes while their organs turn to liquid. That image comes from fiction, and it is wrong. Organs do not liquefy, and dramatic bleeding shows up in a minority of patients. What Ebola does is attack the lining of your blood vessels until they leak. Fluid escapes into places it should never be, and blood pressure collapses. Organs fail from shock, not from anything you would see in a horror film. It behaves less like a monster and more like catastrophic, runaway dehydration, which is why something as simple as fluids and electrolytes saves so many lives. So, 1976, a mission village called Yambuku in what was then Zaire. A new virus had appeared, and a team arrived to work out what it was. A young Belgian scientist named Peter Piot was part of that team. Once they had the virus isolated, they needed something to call it. Somebody suggested Yambuku, after the village where the outbreak was found. Then somebody else pointed out exactly what that would mean. It would permanently attach the name of one small village to a devastating disease. So they looked at a map for something else they could use. They found a nearby river and named the virus after the river, Ebola. Piot admitted years later that they had got even that slightly wrong. The Ebola River is not the closest river to Yambuku. They were exhausted, it was late, and the name stuck. Then the outbreak burned itself out, and the virus disappeared. Here is the problem with a virus disappearing. It cannot wait in the dirt between outbreaks because it is not alive on its own. It needs something living to hide inside. So somewhere out there is an animal carrying Ebola without dying from it. Find that animal and you stop chasing outbreaks and start predicting them. So the hunt began, and 50 years later, it is still going. Researchers have tested thousands of animals across Africa, and they have found traces. Fragments of Ebola's genetic code sitting inside the bodies of fruit bats. They found antibodies too, proof those animals met the virus and survived. Bats have been the prime suspect for 2 decades now. But there is a real difference between finding traces and making a catch. To this day, nobody has ever isolated live, infectious Ebola virus from a free-ranging wild animal. We have fingerprints all over the scene, and we have never once had the suspect in custody. And we know that catch is possible because Ebola has a close cousin called Marburg. Researchers did isolate live Marburg virus from bats. Same family, same hunt, one case closed and one still wide open. And then, in 1989, Ebola turned up somewhere genuinely impossible. That October, a shipment of 100 monkeys arrived at a quarantine facility in Reston, Virginia. That is a suburb roughly 15 miles from Washington, D.C. Within a single month, 29 of those monkeys were dead. Samples went to the Army's infectious disease laboratory at Fort Detrick in Maryland. And under the microscope, the scientists there saw a shape they recognized instantly. A long, curling thread, the signature of a filovirus. Ebola in an office park, down the road from ordinary shops and houses. What followed was a full military and scientific response. The army moved in, and the entire colony of monkeys was euthanized. And then the test results came back on the people who worked there. 4 of those workers had been infected. Ebola virus was in their blood. None of them got sick, not one of them. No fever, no organ failure, nothing at all. What they had found was a previously unknown strain, now called Reston Ebola virus. It is lethal to monkeys, and as far as we can tell, completely harmless to humans. Sit with that, because it is genuinely strange. 2 viruses, close enough to belong to the same small family. One kills a huge share of the people it infects. The other walks into a human bloodstream and does nothing at all. And we still cannot fully explain the difference between them. One detail here I want to be precise about, because it gets exaggerated constantly. Investigators suspected that at Reston, the virus moved between monkeys through the air. Suspected, and between monkeys. It has never been established that Ebola spreads through the air between people. It does not. It spreads through direct contact with bodily fluids. So the search went on, deeper into caves, more bats, more samples. And the whole time, the virus had a second hiding place nobody had checked. In 2014, an American doctor named Ian Crozier caught Ebola in Sierra Leone. He nearly died and was flown to Emory University in Atlanta for critical care. He survived, his blood tested clean, and he was sent home. 2 months later, his left eye began to hurt, and his vision started failing. Doctors drew fluid from that eye and tested it, and it was full of live Ebola. His blood had been clear for weeks, but inside his eye the virus was replicating. The pressure climbed, the inflammation spread, and his iris changed from blue to green. He came close to losing the eye before experimental treatment saved it. Here is why that is even possible. A few places in your body are what immunologists call immune privileged. The eye, the testes, and the central nervous system. Your immune system is held back there because inflammation inside an eyeball would blind you. It is a safety feature, not a flaw. And a virus that reaches one of those rooms is somewhere your defenses cannot storm. Now the part that changes the whole picture. In 2021, Ebola broke out again in Guinea, near an earlier epidemic. Everyone assumed a fresh spillover from an animal, so they sequenced the virus. And it barely differed from the strain behind that earlier epidemic. Viruses mutate constantly as they pass from person to person. This one had hardly changed, which meant it had not been circulating. The explanation that fit the evidence was that it had been sitting inside a survivor for roughly 5 years. Then it woke up and started a brand new outbreak. We had been searching the forest, and some of it was inside the survivors. Which brings us to this summer. In May, the World Health Organization declared the outbreak in the Congo and Uganda an international emergency. By late July, it had passed 2,500 confirmed cases and 1,000 deaths. It is now the fastest-growing Ebola outbreak ever recorded. And there is a specific, frustrating reason this one is hard to stop. We do have Ebola vaccines now, they work, and they were a triumph. But they were built against one species, called Zaire Ebola virus. The outbreak burning through the Congo right now is a different one. Bundibugyo, first identified in Uganda in 2007. 18 years later, there is still no licensed vaccine and no approved treatment for it. So here is where we stand after 50 years of looking. We have a vaccine for one branch of a family whose home we never found. Another branch walked into an American suburb and left 4 infected people unharmed. And the virus can hide inside the people who survive it, quietly, for years. So if it can wait 5 years inside a person, how long could it wait? Put your guess in the comments, and if you learn something, hit subscribe. There is a lot more hiding inside the human body than most people realize. Until then, stay curious.